at Bindley for 5 years. And my colleagues, Amber Yanesh and Johan, are also joining. So they are able to, help with questions also. The idea of this talk is to focus on one technology that has been developed at Purdue in the laboratory of Professor Graham Cooks, which is a high-throughput lipidomic approach. But we are also giving a general overview of what we do and how we interact with the users, how we can support projects and proposals. So that's the idea. We're gonna talk about what we do and who we are at the MPF. The types of lipid analysis platforms we have more than one. And then we're gonna narrow down and focus specifically on this high throughput approach that we use chemical functionalities to guide the profiling. We'll discuss a few applications, and then also how we work through the projects and through the research questions to put together analytical strategy designs for proposals or for experiments that people are interested in performing. And I must say that the place where we are, the Bindley Bioscience Center, we are one of the 9 facilities. It's really an analytical hub for Purdue, where people can come to do proteomics, metabolomics, flow cytometry, genomics, imaging. So it's pretty broad, the types of analysis that you can perform in the facilities in this building. And we're located at the Discovery Park at Purdue. We are 5 people working at the Metabolite Profiling Facility. So this facility has existed for over 10 years. We have been involved in a lot of projects, and usually we run in parallel 30 projects or so at one time. Most of our users are from Purdue, but we do have quite a few from IU, from other states, and also from other countries. Not many, but some of them. And the types of course, we have, instruments and how we perform the services, the diverse methods, but we give full support to people to decide which MS instrumentation, which method, and what workflow of analysis. So we plan together with our users. We try to understand their research and try to suggest the better strategy for their purposes. If they're writing proposals, we can, send them text, revise, or we can even participate as personnel with some involvement if that's useful and it will strengthen the proposal. We also can always provide letters of support stating, that we are ready to do the analysis proposed in the project. Another important component of the facility is training and education. So we understand that the students need hands-on training for their future professional activities. So we have them do sample preparation. If they're using the instrument for a certain period of time, there's some instruments they can get trained on. And we also try to get them as more independently as possible to do data analysis. Also, when your results are generated and you're writing this up for publication, we are supportive also, revising the text, commenting. We don't need to be co-authors. It's a call from the PI, from the team, but we are totally available to contribute with the publication, even if it's just revising or anything. The types of analysis that the facility does are mainly of 3 types. Most of our users are interested in targeted analysis, so they have specific molecules. They're interested in studying dissecting specific pathways. So we have methods that have been established and used for other projects that we can just use the same methods or adapt to their needs easily. But some molecules are still not introduced for our instrumentation. So there is the need of doing some method development. Mostly it's based on literature that, you take the basis and you optimize in the instrumentation that we have for the conditions and for the samples we have. And the other chunk of our work is exploratory lipidomic and metabolomic analysis. So we can generate a list of many metabolites, many lipids, and can also try to identify that things that could be biomarkers of disease or of a certain condition. So this is a video. We're gonna see how is the lab for the. So this was recorded yesterday. This is the entrance. This is inside Bindley. So I'm gonna enter the facility, and so oops, sorry. I wanted to stop the video to talk about the instrument we were seeing. Let's see if I can get that. Okay, so here, when we enter the lab, so this is Bruce Cooper, this is Johan. We're seeing here a UPLC in a cart. So this instrument is shared between two instruments. The one it's hooked into now, it's a triple quadrupole for doing targeted analysis and quantification. People know that also as LC-MS. This is a quite new instrument, and it runs all the time. It's a lot of samples for that instrument. And then when we go to the next row, we're gonna see the QTOF. It doesn't have the UPLC because it's in the other side of the bench. And this big tube here, it's the time of flight tube. That's what gives the instrument the high resolution, the high mass resolution that is used for untargeted metabolomics study. Good. So this is a QTOF. We call this quadrupole time of flight instrument. The next, oh, I'm sorry. I think my mouse is not. So I'm trying to skip, but it's not working very well. Okay, so the next instrument we're gonna see, it's another triple quadrupole. We're going to that, and this triple quadrupole, this is using for the method that we're gonna talk a lot about. That's the untargeted lipid the lipid profile guided by chemical functionalities. And what I'm going to show now is that we don't have chromatography. So, the sample goes directly into the source of the mass spectrometer. We call this direct injection or flow injection. And that's what confers the speed of the approach. This is a gas chromatographer instrument. And his is another triple quadrupole. So that we have quite a few of them because there is a lot of requests. And you see this has chromatography. So it's sample separation and targeted analysis. And this is the newest instrument in the lab. It has a desorption electrospray ionization source for imaging. And this is a CNAPT instrument, is a QTOF as well, is a high mass resolution instrument and has this imaging capability, which is I think a talk by itself because it's a technology, and it has a MALDI source, which is another ionization source, and it has electrospray source also. So it's a quite flexible instrument, and it has another UPLC that also can be used for quantification. This is a mouse brain imaging. We're gonna see some of them also in the presentation, but we're not going to talk much about this technology today, maybe in the future. So in this slide, I just want to give you an overview about the the types of platforms that we have for metabolomics and lipidomics. We have some platforms that don't have chromatography. They're usually known as shotgun approaches. We have a lot of them with chromatography and different types of chromatography depending on the polarity of the molecules. And we have imaging. And all of them have softwares for data processing because mass spec generates a lot of data, and especially when it's exploratory. So there is the need of software and statistics to sort out information and detect what's important, what's relevant to the analysis. Now we're going to switch gears a little bit to lipids, to talk about lipids. And lipids is a special kind of molecule. It's quite different from proteomics. We think proteomics, lipidomics, and metabolomics are in the kind of the same basket, but the molecules that each of this approach deals, it's quite different chemically talking. Proteins are polymers, biopolymers. They're the amino acids and in different positions. So chemically, proteins are very homogeneously in terms of polarity and chemical properties, but the metabolites and lipids, they're not. And the lipids especially, they have a large degree of polarity and diversity in the chemical structures of what we call lipid. So the lipid is very, very broad definition, let's say in this way, in chemical sense. So here is how much we can define the structure of the lipids. Lipids have a lot of isomers, and this can be seen in databases, and this can also be seen in simulation and experiments. The more you want to define the structure of a lipid, so it means the more you want to know which is the position of the unsaturation, which is the we call stereo numbering, where you want to know which fatty acid goes to each carbon of the glycerol backbone, for example, in a phospholipid. The more you want to know, the more you need specific methods and standards and careful analysis to be sure about that. So when we talk about exploratory lipidomics, we are talking about defining some of the structure of the molecule, usually by lipids. So I'm giving here the example of a phosphatidylcholine, which is the structural lipid of membranes, is very commonly detected and studied. And the phosphatidylcholines, they have two fatty acids, and a phosphocholine, what we call this the polar head as well. But usually exploratory lipidomics will give you the two fatty acids, and this underline means you don't know which carbon they are, the position, the stereo numbering position, or you're going to give the sum of the carbons, 20 plus 16, 36, and the sum of unsaturation in both fatty acyl chains for this lipid. So that's what usually exploratory lipidomics will provide you, that level of information. And when we're looking about, hundreds of lipids, and you are in an exploratory objective, that's quite good. And then you can select which lipids are important, are significant, and then you can narrow down the structure. Otherwise it will be a lot of effort, and most of the information will probably not be very relevant. The method that we're talking about today, focusing on this presentation, is called MRM-profiling. It has been developed at Purdue in 2016. And here we're seeing the logo of the Michael J. Fox Foundation, because it was from a project from them that this method came out. So we were trying, this was a project from Professor Cook's lab, and we were trying to profile metabolites or any small molecule present in the cerebrospinal fluid of patients and compared to controls to try to find biomarkers for Parkinson's disease. The diagnosis is a big challenge. Especially early diagnosis of Parkinson's, as most of us know. And we're trying to use very small amounts, and what we were seeing is that it's very diluted, very salty. So when we go to full mass scan, it's very noisy. If we try to do chromatography, you need a lot of samples. So it was not working well for us. So we decided, okay, let's try to move our conception for the method, thinking that, this is a collection of functional groups. We know these functional groups, and the mass spec can profile functional groups for us. So instead of trying to pick up individual molecules and figuring out what they are and trying to connect them using pathway analysis, we were trying to see what's the chemistry that we can detect, and then we can narrow down and we can finish here if these molecules are that we detect doing the functionality turn to be biomarker candidates. And this was in 2014 and 15. But everyone doing metabolomics and lipidomics, I think it's doing research. People who do research with these techniques are very aware of something called the dark matter. And the dark matter means that it's a portion of the metabolome and lipidome that we don't know. It's not in any database, and we're just not aware of the existence of most of these molecules. And it's really a big chunk. It's a problem. So when people say I'm doing lipidomics and metabolomics, it's I think the right way to interpret that is that I'm looking at a portion of the lipidomics and metabolomics. And also, there is not one single technique that can profile the whole metabolomics or the whole lipidomics, because of this chemical variability, structural and chemical variability of the molecules that compose the metabolome and lipidome. They have very different chemical properties and polarities, so it's very challenging, impossible nowadays to have one method that is good for all of them. Some methods will be very amenable to them, but will filter out other molecules. So this is just a fact. And what people are doing is trying to develop, different strategies to cover the metabolome and also improving databases. But meanwhile, I think what is important for us working in this field is to have some analytical possibilities that we can choose from, depending on how many samples we have, what we want to see, how broad we want to see. And here is just the graphical abstract of a manuscript from a group in the University of San Diego. They're really good it's from the Scripps Institute. They're very good with software for metabolomics. So that's what you usually get in a metabolome and lipidome analysis. Most of the ions you detect cannot be assigned because they're not in the database. You have a lot of unknowns. It may be noise, it may be things that are artifacts, but it can be ions that represent lipids that are not in the database. So a small part of them, depending if you do full mass or if you do MS, you can assign them and use them for your biomarker study. But thank you for looking for a biomarker, how much compromise you're doing here. Yeah, how much you are leaving out of your research. So what they did was to try to attribute chemical functionalities to these ions based on specific fragments. And that's more or less what we do, but we do this for profiling the sample, not just for analyzing these ions. But you can improve then a lot. At least you can have something to call these molecules, at least their functional group. And then you can include them in the statistics, and you can go after their structure if that's important for the research. So that's the method that with the concept that's based on what we call the MRM profiling method. And we're so far the only facility to have the expertise to offer this method. And so for this method, we need to use some scan modes in mass spectrometry that can profile the functional groups. So what happens is that when you fragment, when you break down a molecule inside the mass spectrometer, depending on the molecular structure of this molecule, you will see different fragments. And most of the fragments are related to the functional groups present in these molecules, because these functional groups create bonds that are more susceptible to disassemble when you put pressure, what we call collision energy. So this method is based on these scan modes that have been introduced in 1990 by Professor Graham Cooks, and these scan modes are used in mass spectrometry in general. And here is a practical example. I think when we try to go into theory and it turns complicated very fast for so I tried to show some data here just to show how this scans work in the practice. So this is a direct injection experiment. So we're just flushing our sample to the ion source. We have no chromatography. And here what I have is a mixture of different lipids, isotopically labeled lipids, and they are together with the background of the instrument. So anything generates background in the instrument. It can come from the solvent. It can come from some carryover from previous samples. It can come from the environment. So you always have signal in the mass spectrum. So but among the signal, we can see the masses that are expected for some of these standards. And this is what we call full mass scan. If we use this scan that can profile for chemical functionality, we call this neutral loss scan. So what happens is that the instrument is fragmenting all the molecules that come in the first quadrupole, and in the third quadrupole with some mass filter, it's allowing just molecules that lost a specific mass that is related to a functionality. In our case, it's related to the phosphatidylethanelamine. So what's happening, it's losing what we call this polar head, this phosphoamine, and this is causing a shift in the mass. And so if you use this scan, you're gonna pull out the two phosphatidylethanelamines in this whole population here of ions. So you see how much specific you get. So basically, you can rely on the mass spec to do the separation for you internally. It's pulling the class you were interested, the two lipids from the class that were present in the mixture. Another scan is called product ion scan. This scan is specific for ion. So every time we have this solid circle, it means that it's selecting. So the first quadrupole is just selecting one of the lipids, in this case, the 711 here, this PE, and it's fragmenting just it. And everything that fragments it's reading. So it has the original one, and it has the fragment. And it's this loss of 141 that is related to, it's a signature of this phosphatidylethanelamine class. And another scan mode, which is really, really useful, very powerful because it's very sensitive, is called MRM. It's multiple reaction monitoring. In this scan type, the mass spectrometer is just fixed both in an ion and in a fragment of this ion. So for this signal to occur here, this ion was filtered in the first quadrupole, and its fragment coming just from this ion was filtered in the third quadrupole. So this is very specific and also very sensitive, because the mass analyzer is not scanning. It's just looking all the time to the same ion. So this scan is very used when we say LC-MS, targeted analysis. That's the scan we use for, you know, quantifying drugs or so. We use this scan because it's sensitive and it's specific, combined with chromatography. In the case of our technique, we use this scan, a list of these scans to profile the lipids and use this as a profile. Here, I would like to show another, the second scan mode that can profile chemical functionalities is called precursor ion scan. And in the same way as the Nutritional scan, what it's doing is to monitor a specific ion related to a class of lipids. In this case, are the phosphatidylcholines. So all phosphatidylcholines, when submitted to a pressure in the second quadrupole, they will lose their polar head, the phosphocholine. But this has a charge. So instead of having a neutral loss, we will see a fragment. And we have, again, the same mixture, but in the mixture, we had 3 lipids that contain the phosphocholine. So we have 2 phosphatidylcholines and 1 sphingomyelin. So only these 3 are pooled. It is the same mixture. I'm just using the mass spec to filter out this class of lipid. And here's the product ion and the MRM for one of the lipids in the same way we saw in the previous slide. So if we take in consideration the different types of scans that can profile chemical functionalities, and these are, as we just have saw, neutral scan and precursor ion scan, we profile the functional groups in our samples, and we transform this information into the most sensitive and fast scan in mass spectrometry, which is the multiple reaction monitoring, or the MRM, and we scan our samples for these ions that we have found looking at chemical functionalities. We generate a profile. So a list of MRMs that are present in this sample, and this is our profile. And this profile is analyzed by statistics, and we isolate ions, and then it comes to stage 3. Then we use full mass scan, product ion scan, because we want to identify and quantify this molecule. So that's more or less how we work through the different scan modes to generate a workflow that can be exploratory and not very dependent on single molecules or things that are related to databases. Here's a comparison that from the 2 platforms. So as we will see in the case studies, depending on your research, one or the other platform will serve you better. The liquid chromatography, both of them, will have extraction, but of course the LC will have the chromatography separation, with which will select some of the polarities that are present in your sample, and the MRM has no chromatography, so you don't have this filter here, and then you have the database match that will exclude part of the ions that you have been detected without checking if they're imported or not for discriminating your samples. And while here we will have the significant features that are detected by the analysis, so we're not discarding any chemical information that we find. So these are the 2 workflows that we offer in the facility for lipidomics. The most common in the literature is by far LC-MS. Most mass spec companies offer instruments that can specifically do that with dedicated software. So it's very broadly used, and it has a lot of advantages. But also the MRM profiling, the advantage is that you can run more samples in the same time because it's faster. The chromatography always takes time, and also it's guided by chemical functionalities. So of course, they're analytically they are quite different, So it's important to see what kind of information it's needed, how many samples there is in this experimental design. So this is one example. It was a collaboration with Professor Eric Benson at IU, and what he was interested in profiling the lipids present in extracellular vesicles. Extracellular vesicles, they're very tiny samples, so you can get few micrograms of them if you're lucky, and it's very hard to do LC-MS with a microgram amount. Usually you need milligram amount of sample for the LC-MS platform. The QTOF is not the most sensitive mass analyzer, and also the chromatography will dilute, so you need to load a certain amount of sample. And then in this case, we said, okay, let's try the MRM profiling, because we are using the MRM, which is very sensitive scan, a triple quadrupole, which is it's a sensitive mass analyzer. It's low resolution, but it's sensitive. So we have more chances to detect the lipids in these tiny amounts of samples. And here we have detected phosphatidylcholines, ceramides, and some cholesterol esters in those samples. They have been isolated with a kit and from Invitrogen. And what they were interested to see is if lymphocytes that were coming from patients resistant to chemotherapy had different profiles, and they did have a different relationship in the lipids in those extracellular vesicles. And also, if you stimulate or you didn't stimulate, this is lymphocytes in culture, you see the lipid composition of these extracellular vesicles changing. So each point here is one sample, and it represents a panel of lipids. So it's a combination of lipids. It's very well described in the manuscript which lipids we have detected and which ones were important for the discrimination. And here it's an experiment where extracellular vesicles have been recovered from different cell culture types. So we see that, the two cancer cells lines are kind of similar compared to all the other organs. So this is just one application that the MRM profiling has found a fit, and we had interesting results with the technique. This is another example. It's from the Department of Animal Sciences at Purdue. So they were interested in doing a very broad study if how important is the colostrum, this is the mother milk, in cases of obesity. So what they did was to cross-foster mice. So they had the puppies that came from control or from obese mice, and this was nutritional obesity, and they switched some of them. So some mothers have, puppies that did not came from them and were from different background. And we can screen for 10 classes of lipids with MRM profiling. They had a lot of samples, so it was a good fit for them. And what they observed is that the serum of the mothers was quite different in related to the composition of lipids. Sorry, this is difficult to read, but you have a lot of triacylglycerols showing up upregulated in the high-fat diet, which makes a lot of sense. And this transferred to the milk. So the milk was also very upregulated for a lot of triacylglycerols and other compounds that were related to the diet. And here, when they cross-fostered the puppies, was when they look at the serum of the puppies, what was more important, and I think this is expected, it was the serum. It was the milk that they were drinking and not their own metabolism. So their metabolism was completely driven by what they were drinking. And this was expected, but they found a lot of relationship with specific fatty acids that were very upregulated in the high fat diet. So the metabolism changed the fatty acid preference in this kind of this study. So it was really interesting. This is a table showing the diverse studies that we have done over this last 3 years, that with different models, coronary artery disease. This was from Professor Cook's lab, or microbial resistance. Also, a lot of with the acylcarnitines and this is such atopic dermatitis. So how many MRMs have we used for the profiles? How much time we have used to acquire the data for each sample? And what is the performance or agreement? This is a long list, and I have the references related to each of them. So please let me know. I can share with this for, if anyone wants to see the details. So this, I just want to give 2 cases where, we have seen that analytical platform was very important for the results to be significant in this study. So the first one is from Jacqueline Franco and Harm Hogenash from the vet school at Purdue. Jacqueline was doing her PhD thesis studying a mouse model of dermatitis, and they knew that the lipids in the skin were very important for the development of the disease, the composition of the lipids. So they didn't know which lipids they were, and a lot of samples. So we decided to give it a try with the MRM profiling. And the epidermis was a very tiny sample, so it seemed to be a good idea to try that. And I don't know if anyone has a question. Okay, so here we started applying precursor ion scan in the epidermis from these mice. It was parts of the patches of the epidermis, and we have seen here the profile of the phosphatidylcholine and sphingomyelins. Yeah, it doesn't look much different. Some intensities are different, but the ions are pretty much the same. And then here is when we got the profile for the ceramides. The ceramides have an ion that is related to the sphingosine base that they have. So different ceramides have different sphingosine bases. In this in this scan here, in this precursor ion scan, we saw that the control had 10 times less lipids, less signal than the dermatitis one. So we were really impressed on how enriched the skin, the epidermis from the mice with dermatitis were for the ceramides. And this ended up showing, and it was confirmed by LC-MS, that those lipids are really important. They build up as the skin becomes diseased in this model. And Jacqueline recently published also some work doing also the progression, studying how the lipids build up over the progression of the disease. She found some other lipids, cholesterol esters, phosphatidylcholines, and sphingomyelins, and even carnitines. They were also important and were related to male and female, and also to disease progress when there were still no clinical signs of disease. The composition of the lipids in the skin was already changing over time. So it's really interesting. They use some machine learning to define the panel of the lipids in collaboration with Bartek Rajva. The second case of study, it comes from IU, and the idea here was like, okay, we want to do a screening that is broad, but we are also interested in a specific phosphatidylethanolamine that has arachidonic acid and an stearic acid. And then we figure out that this MRM is not the best strategy for that, because with MRM you can detect the P38 carbon and 4 unsaturation in the fatty acid chains, but you are not sure about the composition of the fatty acids, because we're looking at the chemical functionality. We're monitoring the phosphoamine here. So it was not really the technique to answer to this question. So the idea here was to do the liquid chromatography with high mass resolution. And so this is urine samples. And this is here is the total ion current is e to the 7. So all the signal coming from different ions over the course of the chromatographic analysis. And if you extract the ion with the high mass resolution that corresponds to this phosphatidylethaноlamine that has the arachidonic and the stearic acid, you do get a peak. So it was there among many other things that they were interested in profiling together. And we checked with the internal standard, and it matches perfectly the retention time. And also, when you do the high mass resolution analysis of the peak that was in the urine, and the instrument gives the possible molecular formulas that we have, we see that this one here that corresponds to this specific phosphatidylethaноlamine appears with a very high mass accuracy. So and the other ones chemically don't make much sense. So and another information we had was the fragmentation pattern with high mass resolution that the QTOF can do. So you have here is the sample, so you have the same mass as the standard, very similar. And when you do the product ion, when you look at all fragments from this, we see, in the negative ion mode, we see here the arachidonic 303 and 283.2. This is the stearic. So it matches very well. So retention time, fragmentation, and high mass resolution. Next in this research, they were interested in the levels of the PE in this specific PE in the samples, in the 50 samples they had. So it was then Bruce Cooper who did that all these experiments. He came back and he had a calibration curve done with the standard, and then the concentrations could be estimated based on this internal standard. So everything has been done in the LC high resolution mass spec, the QTOF instrument that we have seen in the beginning of our tour. And here I just want to, give you a taste. I will not have time to go through mass spec imaging, but it's really useful. You can map lipids and drugs, metabolites also, in tissue sections or in surfaces. So here you see ink. It looks like it's a number that has not been adulterated, but it has been adulterated because it has two different types of inks in it. And you can detect that using mass spec because it detects the types of molecules present in the samples. So there are a lot of studies using that, especially with the desorption electrospray ionization, but also MALDI. So these are the two main techniques for mass spec imaging. And this is the instrument we have. We saw in the tour is a Synapse XS. Is a very modern instrument for imaging, and it has both MALDI and DESI ion sources. So we're really excited. We're still not fully trained in this instrument, but we're getting it operational. So it's our newest technology in the facility. So as a summary, so that we still have some time to, questions and discuss. And when you do questions, don't feel restricted to, to anything we have talked about. If you have any questions about, small molecule analysis by mass spec, we can totally address them. So as a summary, I'd like to say that the Metabolite Profiling Facility is fully equipped for qualitative and quantitative small molecule analysis. And we are the only ones to offer this high throughput screening of lipids guided by chemical functionalities. It's kind of our specialty. Also, we do a lot of method development. We're available to do that. We are always looking for new technologies that we can use. So we take a lot of user feedback when we write proposals and projects to bring on board new technologies. We do training, we do support. It's part of our mission. We also want to help before sending proposals. You know, it's really good to talk about what is the analytical strategy you're going to propose. Sometimes this has a big impact on how the reviewers receive the proposal. So this is we support before, you know, the experiments can start, and we're pleased to do that. I'd like to thank all my colleagues in the MPF, also many people in Bindley Bioscience with related to administration, other facilities. We interact a lot, and especially Professor Graham Cooks, who for the MRM profiling is our advisor and also driver for the analytical development. Thank you so much. So be happy to take questions.
I think if it's easier, people can also unmute themselves and ask a question. There is one in the chat, I see.
Oh yeah, I see it here. I will jump in. Christina, someone asked, can you measure and compare the size of polyphosphoryl?
That's my question, and then I'm working on bacteria, and my bacteria produce so-called lipoteichoic acid, and then lipoteichoic acid is the anchor is a diacyl glycerol, and then they put the phosphoglycerol, and then there are some variation of the size, and then I just want to measure exact the distribution of the size and compare strain by strain, because that has some correlation with that antibiotic resistance. So I was trying to measure how long that the phosphoglycerol the chain is. And then so far I couldn't find any facility they can do that.
Yeah, so we have studied bacteria resistance using MRM profiling, and it was beta lactamase resistance, and we have challenged them, and we detect a lot of phosphatidylglycerol lipids. But I think we never look at in detail for the structure of these lipids. Maybe if your goal is more structural, maybe chromatography would be helpful. But we could, yeah, maybe we could talk offline that we can see this, you know, the structures and how they would be able to sort them out better, or screen for them if you want to screen first. But yeah, I think we probably can. We detect phosphatidylglycerols in bacteria a lot.
Oh, okay, good. Thanks.
Thank you.
Hi, my question would be about the metabolites and the chemical functions study. So if you do from an in vivo, so would you prefer the serum or plasma?
Usually I prefer, so we do both. I think that the only thing is that the serum has been depleted from proteins, yeah? And some of these proteins have lipids in them. So if you have the plasma, you can, you know, get hand of more lipids that would be excluded from serum. But we do a lot of serum also. I think we're processing now a lot of cow serum for you know, screening. So it depends really if the molecules you were looking at are in serum. If they're small molecules, probably they are, especially metabolites.
Yeah, for example, metabolites or in response to a drug, the metabolites changing. So then serum or plasma, because I mean, for the usual protein analysis, they used to prefer plasma, but I don't know for metabolites or even lipid profile, they may change. So is there any preference depending on, or maybe specific for the what we are looking for?
Yeah. Amber, do you have any experience with that for plasma or serum for metabolites?
So we get both all of the time. It just depends on what makes the most sense for your experiment.
Okay, for the target or general?
Yeah, I mean, there are cases where if there's something in the plasma, a protein that maybe would bind to your metabolite and pull it down, you know, I mean, there are issues you have to think about. It just depends on what you're trying to look for.
Sure. And what is the turnout usually for a metabolite profiling and chemical function? Let's say
So for LCMS based,
yeah.
Bruce would be the contact for that. And what you'll get is basically a list of all the features that it finds, molecular ions that it finds in the system. And he can go out then and blast libraries and return what he can find in the libraries that exist in Metlin and Mona and so forth. So.
Okay, great. Thank you.
Yeah, and he would help you all along the way.
So I saw the information about the contact and everything. Okay, great.
Oh yeah. Sorry, I didn't share that with everyone. I think Jill fixed it. But yeah. So we have a web page. There's an email which I monitor, and if whoever you need, I will punt you along to the right person. So.
Okay, great. Thank you.
Sure thing.
Do you have any other questions? I'm trying to find the chat.
Oh, I see. Yeah, no, I'm looking. So Jill asked about, is it possible to list the established measurements you already have on your website for a new assay? How is the fee charged? So I can answer the fee part. So we bill per hour. So it all depends on how long it takes for us to put the method together and validate it and confirm it. It's really hard for us to say from the outset, oh, it'll take 3 hours to do. We don't really know until we get into it. And then we need to get a lot of information from you. We need to know how many samples you're going to have, what kind of sample you're going to have, what your analytes are. So we usually meet with everyone and sort of chat and go through it all and go from there. We try to give you a rough estimate once we have more information. Yeah, everything's on the website. And yeah, Christina, that was a question I had. You, in one of your slides, you said you had roughly 10 classes. How many classes of molecules are you looking for now, or can you look for?
So we do with the MRM, because of the chemical functionality thing, we target it to some classes. When you do the LC with high mass resolution, you don't know, it's whatever comes out. So, but with MRM profiling, we have the phospholipids. So we have phosphatidylcholines, ethanolamines, inositols, glycerols, and serines, the 5 phospholipid classes. And then we profile cholesterol esters, acylcarnitines, ceramides, triacylglycerol, and maybe I'm missing one.
It's okay. Put you on the spot.
But it's and some people have specific, like, I want to monitor this metabolite, or I want to monitor specific things, and we can try. We say, okay, let's start with a test, see if you can have signal. And one important thing about MRM profiling is that we need to look at it as exploratory. So what the attribution we're giving from this technique, it's our best guess, it's tentative. So it's meant to be a kind of umbrella, and then you detect, okay, this lipid seems to be really interesting, and then it's interesting to validate by liquid chromatography with the standard and everything. So we see this, but then so we need just to be careful of having this having this in mind, that it's exploratory. So we need to confirm if it's really, really important.
Right, and we do that all the time. We'll get certain classes that people are interested in, and we'll use the dreaded LC column to do the separation, and we buy standards and we confirm everything that way. That is also an option. Any other questions?